DNA is the blueprint of life, carrying the instructions needed for growth and development. Before a cell divides, it must make an exact copy of its DNA. This process is called DNA replication, and in eukaryotes, it happens inside the nucleus during the S phase of the cell cycle. The DNA molecule is a double helix made up of two complementary strands. Each strand acts as a template to produce a new complementary strand. Because each daughter DNA contains one old strand and one new strand, DNA replication is called semi-conservative. Unlike prokaryotes, eukaryotic chromosomes are very large and linear. Replication begins at multiple origins of replication, which speeds up the process. From each origin, replication proceeds in both directions, forming replication bubbles that eventually merge. At each replication origin, the enzyme Helicase unwinds the DNA double helix by breaking hydrogen bonds between base pairs. This creates a replication fork. Proteins called single-strand binding proteins attach to the separated strands, preventing them from rejoining. Another enzyme, Polymerase, relieves the tension created ahead of the fork. But DNA polymerases cannot start replication on their own. They need a starting point. Here, Primase lays down short RNA primers that provide a free 3O group for DNA polymerase to begin synthesis. The main enzyme, DNA polymerase, now adds new nucleotides complementary to the template strand. On the leading strand, synthesis is continuous in the 5-to-3 direction. On the lagging strand, synthesis is discontinuous, forming short fragments called Okazaki fragments. Once the fragments are built, the RNA primers are removed by enzymes like RNase H-DNA polymerase, then fills in the gaps with DNA nucleotides. Finally, DNA Ligase joins the Okazaki fragments, sealing the nicks to form a continuous strand. At the ends of eukaryotic chromosomes lie special structures called telomeres. They protect coding DNA from being lost during replication. An enzyme called telomerase extends telomeres, ensuring that essential genetic information is not lost. In the end, the result is two identical DNA molecules, each with one old and one new strand, ready to be passed on to the daughter cells. This ensures genetic continuity from one generation of cells to the next. To summarize, the key steps in eukaryotic DNA replication are: 1. Multiple origins of replication form. 2. Helicase unwinds the DNA helix. 3. Primase lays down RNA primers. 4. DNA polymerase builds new strands. 5. Okazaki fragments are joined by Ligase. 6. Telomeres are maintained by telomerase. DNA replication is fast, accurate, and highly regulated. The very reason life continues smoothly across generations.